Think about it for a second. Your heart. It's this tireless engine, right? Quietly performing over 100,000 beats every single day without you even thinking about it. From when you're sleeping soundly to, an all-out sprint, it's constantly adapting. It's just this marvel of biological engineering pulsing life through your body. It's truly astounding when you stop and consider it.
It really is, isn't it? That consistent, unwavering rhythm.
Oh,
Just a testament to incredible design. A true masterpiece, like you said.
absolutely. And today, that's exactly what we're doing, taking a deep dive into that masterpiece, the fascinating world of cardiac muscle. We're aiming to go beyond the basics here.
Beyond.
Yeah, really peeling back the layers, uncovering the hidden mechanisms, the sort of cellular choreography, and the sophisticated ways your heart fine-tunes its power. Think of this as your shortcut to understanding cardiac muscle like the true professionals in the field. Our goal is mastery here.
Exactly. Our mission today is precisely that, to arm you with a really thorough, deep understanding. We'll explore how these specialized cells are organized, how a tiny electrical signal translates into such a powerful pump,
Right.
what makes cardiac muscle unique compared to, say, skeletal muscle, and critically, how it responds to things like stress, stretch, and even subtle genetic mutations.
So by the end of this, you'll have a grip on the heart's fundamental biology and the conditions that affect it, truly at an expert level.
Yes, the plan.
Okay, so let's start at the very foundation then. The incredible cells that make up this muscle. What's the basic blueprint of these cardiomyocytes, is that right? And what makes their organization so unique?
Well, these unsung heroes, the cardiomyocytes, they're actually relatively small cells, maybe 10 micrometers in diameter, 100 long. But what truly sets them apart, as you hinted, is their interconnectedness.
Ah.
Unlike skeletal muscle cells, which are more independent, cardiac cells form what's known as an electrical syncytium.
Okay, electrical syncytium. That sounds important. It must be critical for that synchronized action you mentioned.
Absolutely critical. It's all about these remarkable structures called intercalated discs. They literally glue cardiac cells together end to end.
Glue them together. Okay.
And within these discs, you find two main types of connections. First, you've got the mechanical ones, things called fascia adherens and desmosomes. Think of them like heavy-duty rivets.
Rivets. Okay, so they stop the cells tearing apart.
Precisely. They prevent the cells from pulling apart under the immense contractile force of each heartbeat. They provide that physical tethering.
Got it. And then there are the electrical connections, too.
Exactly. That's where gap junctions come in. These are like microscopic direct wires between the cells.
Direct wires.
Yeah, they allow action potentials, those tiny electrical signals we talked about, to propagate really quickly and efficiently throughout the entire heart muscle.
Okay. So it's this combination then, the mechanical rivets and the electrical wires.
Right. This combined mechanical and electrical syncytium means that a single action potential, the one starting way up in the sinoatrial node, the heart's natural pacemaker, can just sweep through the entire heart almost instantaneously.
And that's the genius of it, isn't it? This super rapid, widespread signal ensures that synchronized wave-like contraction needed for pumping blood, but also, equally important, synchronized relaxation so the chambers can refill properly. It's like an all-or-nothing system for every beat.
Exactly right. Without that perfect synchronization, the heart just couldn't pump effectively. It would be chaos.
Makes sense.
Now, despite these really unique cell-to-cell connections, if you actually look inside a cardiac muscle cell, you'll see it looks striated.
Striated, yeah, like skeletal muscle, those stripes.
It has those repeating light bands, the I bands, and dark bands, the A bands. Very similar appearance under a microscope.
Right, and the Z line runs through the middle of the I band, anchoring the thin filaments, and that ultimately defines the sarcomere, the basic contractile unit. This is where the real action happens, isn't it?
This is absolutely where the magic happens. The sarcomere, the region between two adjacent Z lines, and inside it, you've got your thin filaments made of actin, tropomyosin, and troponin extending into the A band.
Okay.
And then the thick filaments composed of myosin extending from the center of the sarcomere out towards the Z lines.
And the myosin filaments are arranged in a specific way for pulling, I remember.
Yes, very cleverly organized. They associate tail to tail right in the center, and then head to tail as they extend outwards.
Yeah.
This positions the myosin heads perfectly to grab onto and pull the actin filaments inward during contraction. It's all about that precise pulling action.
It's incredibly precise engineering at a molecular level, and to make sure that precision holds up, the sarcomere also relies on this intricate network of supporting or scaffolding proteins.
Like microscopic engineers, as you said.
Yeah. Maintaining the structure for optimal function. We have things like meromyosin and C protein organizing the thick filaments, nebulin and trypamodulin working on the thin ones, and alpha-actinin anchoring things at the Z line. But among all these, one really stands out, doesn't it? Structurally and functionally. Titin.
Titin. Yes, the elastic giant. It's a remarkably large protein, truly enormous, and it's elastic. It tethers those thick myosin filaments to the Z lines.
So it's partly structural, preventing overstretching of the sarcomere itself.
It definitely does that. But what's really fascinating, and something researchers are digging into more, is that titin isn't just passive. It seems to act as a stretch sensor.
A sensor? How so?
It appears to participate in cell signaling, potentially modulating protein synthesis in response to mechanical stress or load. It's communicating with the cell.
So it's not just a passive spring absorbing tension, it's actively involved in how the heart adapts. That's amazing.
It is. And this signaling role has real clinical relevance. Genetic defects in titin, tendinopathies as they're called, can lead to muscle atrophy in both the heart and skeletal muscles, contributing to cardiac dysfunction and certain muscular dystrophies.
Wow.
And its elasticity, that springiness, also significantly contributes to the heart's ability to increase its force when it's stretched. A really critical mechanism we'll definitely get back to.
Okay, titin. Definitely one to remember. Now, speaking of structure, you mentioned earlier that the heart actually has more connective tissue than skeletal muscle. That seems a bit counterintuitive, maybe? Why is that so crucial?
It's a critical, potentially life-saving difference. While connective tissue everywhere helps prevent muscle rupture, in the heart, its abundance serves another vital function.
Yeah.
It prevents overstretching of the entire organ.
The whole heart itself.
Yes. If you look at length tension curves, cardiac muscle shows this dramatic, steep increase in passive tension resistance when it's stretched beyond its normal resting length. Skeletal muscle, on the other hand, can be stretched much further before that passive tension really ramps up.
So the heart is much less stretchy in a way. It relies heavily on that connective tissue wrapper to keep it contained.
It absolutely does. It's a fundamental safeguard. Think about periods of increased venous return, like during an intense exercise, when blood flow back to the heart can increase fivefold.
5 times, that's huge.
It is. But even with that massive influx, the heart manages to pump it all out with only relatively minor changes in its volume, maybe less than a 20% increase in end diastolic volume. The connective tissue provides that limit, that boundary.
And what would happen if it did overstretch?
Catastrophe, basically.
Yep.
If the heart overstretched, its ability to contract would actually decrease significantly because the actin and myosin filaments wouldn't overlap optimally anymore.
The sarcomere function breaks down.
Exactly. That would lead to insufficient pumping, blood backing up, increased venous pressure, and potentially severe conditions like pulmonary edema fluid in the lungs. So that extra connective tissue is a built-in safety net against catastrophic failure.
That makes perfect sense, a vital protective mechanism. Okay, now let's zoom back inside the cell and talk about the heart's internal plumbing. The sarcoplasmic reticulum, the SR, and the T-tubules.
Right, so the SR is that internal membrane network surrounding the myofibrils, the contractile elements. It's similar to skeletal muscles SR, but interestingly, it's generally less dense and developed in the heart.
Less dense, okay. And the T-tubules.
Those are the invaginations of the outer cell membrane, the sarcolemma. They dive deep into the cell, carrying the action potential inwards, close to the SR.
And there are key differences here compared to skeletal muscle too, aren't there? Structural differences with functional consequences.
Yes, subtle but profound differences. In cardiac muscle, the T-tubules are positioned at the Z lines. In skeletal muscle, they're at the ends of the I bands, near the AI junction. Also, the actual connections between the T-tubules and the SR membrane are fewer and maybe less organized in the heart.
Fewer connections.
Which leads to a distinct structural arrangement. Cardiac muscle typically has dyads. That means a T-tubule membrane is associated with just one adjacent SR membrane.
Just one, a dyad.
Whereas skeletal muscle famously has triads, where a T-tubule is sandwiched right between two terminal cisternae of the SR.
Dyads versus triads, okay.
And this structural distinction, dyad versus triad, really underpins a key functional difference in how calcium is handled, which brings us nicely to the spark of life itself, excitation-contraction coupling in the heart.
Right, this is where the heart's intrinsic rhythm originates. We know cardiac muscle is involuntary, it has its own pacemaker. How exactly does it set this relentless beat?
Well, that rhythm starts in the sinoatrial node, the SA node. There are specialized cells there that spontaneously depolarize. They generate their own electrical signals, action potentials, setting the basic heart rhythm.
So they just fire on their own?
They do. Now, other heart cells can also spontaneously depolarize, but the SA node cells do it the fastest, so they normally drive the beat. They're the dominant pacemaker.
Got it. And once that first signal fires from the SA node?
It spreads incredibly fast. Thanks to those gap junctions we talked about and some specialized conduction pathways, the action potential can sweep through the atria, the top chambers, in about 70 milliseconds.
Wow.
And the entire heart, ventricles included, within about 220 milliseconds after the SA node initiates the beat. This rapid, widespread conduction is key.
Because it ensures that nearly synchronous contraction we mentioned earlier.
Exactly. Vital for effective pumping. And this is a huge difference from skeletal muscle, right? Where you recruit motor units one by one or in groups to grade the force. The heart basically goes all in with every beat.
All or nothing for the whole muscle, yet it still needs to modulate its strength somehow.
Precisely. And to achieve that modulation, calcium plays an absolutely crucial and unique role. Here's a fundamental distinction. Cardiac muscle absolutely requires extracellular calcium to contract.
Requires it? You mean it won't contract at all without calcium from outside the cell?
Pretty much. If you remove extracellular calcium, contractions cease almost immediately, even if you keep electrically stimulating the muscle. Skeletal muscle, by contrast, can keep contracting for a while without external calcium because its internal calcium stores within the SR are sufficient on their own.
That's a massive difference. The heart is essentially plugged into the external calcium supply for every single beat.
You could think of it that way.
Yeah.
And related to this, cardiac muscle action potentials are much, much longer than skeletal muscle ones. We're talking 150 to 300 milliseconds compared to maybe 5 milliseconds in skeletal muscle.
Okay, why so long? What causes that prolonged electrical signal?
That prolonged plateau phase of the cardiac action potential is primarily due to a slow inward current of calcium ions. This current flows through specific channels called L-type voltage-gated calcium channels. The main type is Kv1.2, located in the sarcolemma, and importantly in the T-tubules.
The L-type channel. So calcium flowing in keeps the cell depolarized longer.
Exactly. And here's where the trigger calcium concept becomes absolute central. The relatively small amount of calcium that actually enters the cell through these Kv1.2 channels during the action potential isn't enough on its own to cause full contraction.
Not enough. Then what does it do?
It acts as a critical trigger. This incoming calcium stimulates the release of a much, much larger amount of calcium from the internal stores, the sarcoplasmic reticulum.
Ah. So this small initial spark of calcium coming in from the outside ignites a much larger internal explosion of calcium release.
That's a great way to put it. And the channels on the SR that release this huge secondary waves of calcium are called ryanodine receptors, specifically the RYR2 isoform in the heart. They're concentrated right near the T-tubules, where the trigger calcium enters.
Does the trigger calcium binds to or activates these RYR2 channels?
Correct. It's a process called calcium-induced calcium release, or CICR. And this mechanism really defines the core difference in excitation-contraction coupling between heart and skeletal muscle.
Okay, explain that difference again. Electrochemical versus electromechanical.
Right. In cardiac muscle, it's electrochemical coupling. The voltage change opens the Kv1.2 channels, calcium influx occurs, and that chemical signal, the calcium ion itself, triggers release from RYR2.
Voltage calcium influx, day signal calcium release. Electrochemical.
Exactly. In skeletal muscle, however, it's considered electromechanical coupling. The voltage change causes a physical conformational change in its L-type channel, Kv1.1. This physical change is directly transmitted, likely through protein interaction, to the skeletal muscle ryanodine receptor, RYR1, causing it to open and release calcium from the SR.
So skeletal muscle uses more of a direct physical push, whereas the heart uses that small calcium spark.
Precisely. Skeletal muscle doesn't strictly require calcium to enter from the outside to trigger SR release for a single twitch. The voltage change and physical link are enough. The heart absolutely depends on that trigger calcium influx. It's a fundamental difference in how they initiate contraction.
That is a critical distinction. Okay, so calcium floods the cell. When it comes to the actual contraction mechanism, the sliding filaments, that part is more familiar, right? But with a twist.
Yes, the downstream events are quite similar. Like skeletal muscle, cardiac muscle contraction is regulated by the thin filaments. That means an increase in intracellular calcium is the necessary switch.
The calcium switch.
Right. At low calcium levels, let's say below 50 nanomoles per liter, tropomyosin lies in a position on the actin filament that blocks the sites where myosin heads want to bind.
Blocking the binding sites.
But when calcium levels rise inside the cell, calcium ions bind to a specific subunit of troponin called troponin C. This binding causes a conformational change in the whole troponin complex, which then physically pulls tropomyosin out of the way, exposing those myosin-binding sites on actin.
Unmasking the sites.
Exactly. And as long as that cytosolic calcium remains elevated, myosin heads will bind to actin, go through their power stroke, pull the thin filaments, and contract the muscle cell.
And the myosin crossbridge cycle itself, that four-step dance, is identical to skeletal muscle.
That part is identical. You have myosin, which is energized or cocked, binding to actin once the site is exposed. Then it releases inorganic phosphate, triggering the power stroke, the pulling action. Then a new ATP molecule binds to the myosin head, which causes it to detach from actin. And finally, the myosin head partially hydrolyzes that ATP, using the energy to recock itself, ready for another cycle if calcium is still present and binding sites are open.
Bind, pull, release, recock. Got it.
That's the universal mechanism. But here's the crucial twist for cardiac muscle, and it relates back to that syncytium idea and the need to modulate force without recruiting more cells.
Right, because all the cells contract together in one beat.
Exactly. So recruiting more muscle cells isn't an option like it is in skeletal muscle. And tetany, a sustained fused contraction, would be lethal for the heart. It needs to relax to fill.
Can't have the pump seize up.
Absolutely not. So cardiac impulse has evolved a different way to modulate its force. It does so by varying the amplitude of the intracellular calcium transient.
Varying the calcium level?
Yes. A stronger signal, like from sympathetic stimulation, leads to a bigger surge of calcium released from the SR. This bigger surge activates more troponin C molecules, exposes more myosin binding sites, allows more cross bridges to form simultaneously.
And therefore generates more force.
Precisely. A weaker signal means a smaller calcium surge, fewer active cross bridges, and less force. It modulates force by grading the amount of calcium released per beat, not by recruiting more cells or summing contractions. It's a brilliant solution.
That really is clever. Instead of more workers, make the existing workers work harder by giving them a stronger signal.
That's a perfect analogy.
Okay, so the contraction happens. Now the heart needs to relax efficiently to prepare for the next beat. This is the art of letting go, clearing that calcium out. How does that happen?
Indeed, relaxation is just as active and critical as contraction. It requires rapidly decreasing that cytosolic calcium concentration back down to resting levels. A major player here is the SERCA2 pump.
SERCA, the pump on the SR membrane.
That's the one. The sarcoplasmic endoplasmic reticulum K plus AVNAC ATPase. It actively pumps calcium ions from the cytosol back into the SR using ATP. Similar function to SERCA1 in skeletal muscle.
Pumping it back into storage.
Right. But remember that trigger calcium, with the calcium that entered from outside the cell to initiate the whole process.
Ah, yeah, the heart needs external calcium.
So simply pumping calcium back into the SR isn't enough over the long term. The cell also needs mechanisms to actively extrude that trigger calcium completely out of the cell to prevent a gradual buildup, which would be toxic.
Okay, so how does it get calcium out of the cell?
There are two main mechanisms on the sarcolemma, the outer membrane. The most important one quantitatively is the sarcolemmal 3Na plus NECara plus amproporter, often called the sodium-calcium exchanger, or NCX.
3 sodiums in for 1 calcium out.
Exactly. It cleverly uses the electrochemical gradient for sodium, which is typically high outside the cell and low inside, to drive calcium out against its concentration gradient. It swaps 3Na plus ions moving in for 1K plus ion moving out.
And you mentioned it's electrogenic, creates a current.
Yes, because it moves 3 positive charges, Na plus, in for only 2 positive charges, say K plus out. There's a net movement of 1 positive charge into the cell with each cycle. This generates a small inward depolarizing current that can actually influence the cell's electrical activity, especially under certain conditions.
Interesting. So that's the main extruder. What's the other one?
There's also a sarcolemmal K plus pump, or PMCA, plasma membrane K plus ATPase A. This one directly uses ATP to pump calcium out of the cell. It's generally thought to play a smaller role than NCX in bulk calcium removal during relaxation, but might be important for fine-tuning resting calcium levels.
So SERCA pumps it back into the SR, NCX swaps it out for sodium, and the PMCA pump directly pushes it out using ATP, a coordinated cleanup crew.
A highly coordinated and essential cleanup crew. They all work together to swiftly drop cytosolic calcium, allow troponin to release calcium, let tropomyosin block the binding sites again, and thus cause relaxation.
Now, I remember reading something about the difference between free calcium and total calcium needed. Can you clarify that? It seemed like a huge amount of calcium was involved overall.
That's a really important point for understanding the scale. You're right. While the free ionized calcium concentration only needs to rise from a resting level of maybe 50, 100 nanomoles per liter up to perhaps 600 nanomoles per liter to get significant force.
That sounds like a relatively small increase in concentration.
In terms of free calcium, yes. But the cell is packed with proteins that bind calcium, not just troponin C, but also calmodulin, parvalbumin in some species. The pumps themselves bind it transiently. So to achieve that required increase in free calcium, a much larger total amount of calcium needs to be mobilized and enter the cytosol.
Because most of it immediately gets bound up by something.
Exactly.
Okay.
Estimates suggest that the total myoplasmic calcium concentration might increase by something like 70 micromoles per liter during a strong beat. That's orders of magnitude higher than the free concentration change. It highlights how much calcium is actually fluxing.
70 micromoles per liter. That's a huge amount moving around. And how much of that comes from the SR versus influx from outside?
It varies a bit by species, but in many mammals, including rabbits, dogs, cats, guinea pigs, and importantly humans, the SR uptake and release cycle accounts for roughly 70% of that intracellular calcium transient.
So the SR is the main player, but that 30% from outside is still significant.
Absolutely. That 30% represents the trigger calcium influx via L-type channels, and also potentially some contribution from reverse mode NCX under certain conditions. But it's crucial. Interestingly, in species like rats and mice, they rely much more heavily on the SR, maybe around 92% SR reuptake.
So there are species differences in the balance. Good to know.
Definitely. It highlights the diverse strategies nature uses, but the fundamental principles of trigger calcium and SR release hold true.
Okay, so we've got the contraction, the relaxation, the calcium handling. Now, how does the heart actually fine-tune the strength of its beat on a moment-to-moment basis, beyond just the basic rhythm set by the SA node?
Right, modulation of force. This involves both intrinsic mechanisms, things inherent to the heart muscle itself, and extrinsic control, mainly from the nervous system and hormones.
And we already established it can't use recruitment or tetany.
Correct. That long refractory period, thanks largely to those L-type calcium channels, prevents tetany. So the heart's primary strategy for modulating force is, as we discussed, altering the amplitude of that intracellular calcium transient, basically, controlling how much calcium gets released with each beat.
Okay. Now, in a lab, you could change the external calcium concentration to see force change. Lower external count on plus LREO, less trigger cal, plus is, less SR release, weaker contraction.
Yes, that works beautifully in vitro. Decreasing extracellular calcium reduces contraction force without really changing the duration much because the kinetics of circa and the extrusion mechanisms aren't directly affected.
But in a living person, the body doesn't typically regulate heart force by fiddling with blood calcium levels, right? That's kept pretty constant.
You're absolutely correct. While severe abnormalities in blood calcium can affect heart function, it's not the normal physiological way the body fine-tunes beat strength. The most powerful and common way this happens in vivo is through sympathetic nervous system stimulation.
Ah, the fight or flight response.
Yes, exactly. Triggered by excitement, fright, exercise, any situation demanding higher cardiac output. This involves the release of norepinephrine directly onto heart cells from sympathetic nerve endings, or epinephrine circulating in the blood from the adrenal medulla.
And these hormones, these catecholamines, bind to specific receptors on the heart cells.
Yes, they primarily bind to adrenergic receptors, specifically DOA1 receptors on the cardiomyocytes. This binding kicks off a crucial intracellular signaling cascade.
The Simperi pathway.
Precisely. Receptor activation stimulates an enzyme called adenylate cyclase, which converts ATP into cyclic AMP, or CAN-MP. CAMP then acts as a second messenger, activating another key enzyme called protein kinase A, or PKA.
Protein kinase A, the A for CAMP dependent.
Right, and PKA then goes around phosphorylating, adding phosphate groups to several key target proteins involved in calcium handling and contraction.
Okay, what are the main targets?
There are several, but three are particularly critical. First, the L-type voltage-gated calcium channels, Kb1.2, themselves get phosphorylated.
What does that do?
Phosphorylation increases the probability that these channels will open in response to depolarization, and perhaps how long they stay open. This means more trigger calcium enters the cell during the action potential.
More trigger, okay.
And interestingly, there's often an A kinase anchoring protein, an AK, physically tethering PKA, right near the L-type channel, making this phosphorylation very efficient and localized.
Wow, targeted delivery. What's the second target?
The second key target is a small protein called phospholamban, or PLN, which is associated with the Circa pump in the SR membrane.
Phospholamban, PLN.
Normally, in its unphosphorylated state, PLN acts like a brake, inhibiting Circa activity. But when pKa phosphorylates PLN, this inhibition is relieved. The brake comes off Circa.
So Circa pumps calcium back into the SR much faster.
Much faster and more effectively. This allows the SR to accumulate more calcium between beats before it gets extruded by NCX or PMCA, so the SR stores become superloaded.
Superloaded SR stores, got it. And the third target?
pKa also phosphorylates troponin I, TNI, one of the subunits of the troponin complex on the thin filament. This phosphorylation actually decreases the sensitivity of the myofilaments to calcium.
Wait, decreases sensitivity? That sounds counterintuitive if you want a stronger contraction.
It does at first glance. But think about the overall effect. You've got more trigger calcium coming in, and crucially, a superloaded SR ready to release a much larger amount of calcium when triggered. This massive increase in calcium release overwhelms the slight decrease in sensitivity caused by TNI phosphorylation.
Ah, okay, so the sheer amount of calcium available dominates.
Exactly. The net effect is a significantly stronger force of contraction due to more cross bridges forming. This is called positive inotropy.
Positive inotropy, stronger beat. Makes sense.
But that's not all. Remember the phosphorylated phospholamban speeding up Circa?
Yes. Faster pumping back into the SR.
That leads to a faster rate of relaxation. The calcium is cleared from the cytosol more quickly, allowing the muscle to relax sooner. This is called positive lusitropy.
Lusitropy, faster relaxation. Why is that important?
It's incredibly important, especially when the heart rate increases, which also happens during sympathetic stimulation that's positive chronotropy, a direct effect on the SA node. Faster relaxation allows more time for the ventricles to fill with blood between these more frequent beats. If relaxation didn't speed up along with the rate, filling would be compromised.
Ah, so it keeps filling efficient even at high speeds. Clever.
It's a beautifully coordinated response: stronger contractions and atrophy, faster relaxation, lusitropy, and increased heart rate, chronotropy. The overall result is a heart that can pump more blood, more forcefully, more often, and fill effectively between beats, perfectly adapted for fight or flight or intense exercise.
A complete system upgrade on demand. Are there any other, maybe newer players in this regulation game? You mentioned phospholamban.
Well, yes. Quite recently, researchers identified a very small peptide, only 34 amino acids long, called dwarf. It seems to be encoded by what was previously thought to be non-coding RNA.
Dwarf, what does it do?
It appears to directly interact with Circa and can increase Circa's affinity for calcium. It might do this by actually competing with phospholamban and displacing it from Circa.
So it could boost SR calcium uptake, similar to phosphorylating phospholamban, but through a different mechanism.
Potentially, yes. It represents another layer of regulation, improving SR function and myocyte performance. It's an exciting area, showing we're still discovering new details about how this intricate system is fine-tuned.
Fascinating. But sometimes this fine-tuning goes wrong, especially with genetic mutations. You mentioned the ryanodine receptor, RYR2, earlier. Mutations there can cause serious problems, right, like CPVT.
Yes. Mutations in the cardiac ryanodine receptor gene, RYR2, are a major cause of a dangerous inherited arrhythmia syndrome called catecholaminergic polymorphic ventricular tachycardia, or CPVT.
Catecholaminergic, meaning it's triggered by adrenaline or similar stress hormones.
Exactly. Patients with CPVT are often fine at rest, but during exercise or emotional stress, when sympathetic activity increases, they can develop life-threatening ventricular arrhythmias.
What's happening at the channel level with these mutations?
These mutations typically make the RYR2 channel leaky, particularly during diastole relaxation when it should be tightly closed. They cause spontaneous inappropriate calcium release, or leak, from the SR.
Leaky channels.
So during stress, you have the normal sympathetic drive increasing calcium cycling, plus this underlying leak from the mutated RYR2. This combination leads to an overload of calcium in the cytosol during diastole and can also cause oscillations in SR calcium load.
And how does that trigger an arrhythmia?
That elevated diastolic calcium does a couple of things. One key effect is that it abnormally activates the sodium-calcium exchanger, NCX, in its forward mode, pumping Km++ out and A+ in. Remember, NCX is electrogenic.
Right, it brings positive charge in.
So this abnormal NCX activity generates an inward current during diastole. If this current is large enough, it can push the cell membrane potential back up towards the threshold for firing another action potential. These are called delayed after depolarizations, or DADs.
An extra unwanted beat triggered by the calcium overload and NCX.
Precisely. If these DIAs reach threshold, they can trigger ventricular tachycardia, which can degenerate into fibrillation. That's the danger of CPVT.
Terrifying. How is it treated?
Because it's triggered by catecholamines, the first line of treatment is usually anti-adrenergic therapy, typically beta blockers, to blunt the sympathetic response and reduce the calcium cycling during stress. For patients who don't respond adequately, an implantable cardioverter defibrillator, ICD, might be necessary to shock the heart back into a normal rhythm if a dangerous arrhythmia occurs.
A stark example of how disrupting calcium homeostasis can have lethal consequences. Okay, let's shift gears to another fundamental control mechanism. Stretch. The Frank-Starling law of the heart.
Ah, yes, a cornerstone of cardiac physiology. This is an intrinsic mechanism, meaning it's inherent to the heart muscle itself, independent of nerves or hormones. The law basically states that the more the heart muscle is stretched before contraction, up to a point, the more forcefully it will contract.
More stretch equals more force. When does this normally happen?
It happens whenever the volume of blood returning to the heart increases what we call increased venous return or preload. This occurs during exercise, for example, or even just when you lie down or if the heart rate slows, allowing more time for the ventricles to fill between beats.
So the heart automatically adjusts its output to match the input.
Exactly. It ensures that, within limits, the heart pumps out whatever volume of blood it receives. It's a crucial beat-to-beat regulator of stroke volume, matching output to input and also balancing the output of the right and left ventricles.
And you mentioned the passive tension earlier from connective tissue and tighten. That plays a role here too.
It does. As the heart stretches during increased filling, that passive tension rises significantly. This helps prevent overstretching and keeps the sarcomeres operating within their optimal length range. Remember, cardiac muscle has much higher passive stiffness than skeletal muscle.
Right, the protective effect. And this stretch-induced force increase, it happens over a specific range of muscle lengths.
Yes, it's particularly steep and important over the physiological range of sarcomere lengths in the heart, roughly from about 1.6 up to maybe 2.3 micrometers. This creates what's called steep length-dependent activation.
Okay, so what's the mechanism behind the Frank-Starling effect? Why does stretching the muscle make it contract harder? Is it just better filament overlap, like in basic muscle physics?
That's part of it, but actually not the dominant factor in the heart, especially at the typical operating lengths. Changes in myofilament overlap probably account for maybe 40% or less of the effect.
Less than half. So what's the main reason?
The major contributor, accounting for about 60% or more of the increased force, is an increase in the calcium sensitivity of the contractile proteins.
Increased sensitivity again. So the same amount of calcium causes a bigger response when the muscle is stretched.
Exactly. When the sarcomere is stretched, the myofilaments, particularly troponin C, become more sensitive to the calcium that is released during the normal excitation-contraction coupling process. More cross bridges form for a given level of calcium.
Why does stretch increase calcium sensitivity? What's happening there?
The precise molecular mechanism is still debated and likely involves multiple factors. Changes in the lattice spacing between thick and thin filaments might play a role, bringing them closer together. Also, conformational changes in key proteins like titin, troponin T, and perhaps myosin-binding protein C, which are all linked structurally, are thought to contribute to this length-dependent tuning of calcium sensitivity. It's an area of active research.
So it's less about just overlap, more about tuning the response to the existing calcium signal.
Very interesting. Okay, we've covered structure, contraction, relaxation, regulation. What about the fuel? Sustaining this engine requires a massive amount of energy. Cardiac muscle metabolism.
Absolutely critical. That myosin cross-bridge cycling burns through ATP constantly. So those ATP pools need to be replenished continuously, beat after beat, second after second.
Where does all that ATP come from?
The heart is an aerobic powerhouse. Its primary source of ATP is aerobic metabolism, cellular respiration, primarily through the oxidation of fatty acids, but also carbohydrates like glucose and lactate. It's highly dependent on a constant supply of oxygen.
Fatty acids are a major fuel source then.
Yes, under normal resting conditions, fatty acids are often the preferred fuel, but the heart is quite flexible and can readily switch between fats, glucose, lactate, even ketone bodies depending on availability and demand.
Is there any backup system, like in skeletal muscle?
Hmm?
Creatine phosphate? There is a pool of creatine phosphate, yes. It acts as a small, immediate buffer. The enzyme creatine kinase can quickly transfer a phosphate group from creatine phosphate to ADP to regenerate ATP. But this pool is relatively small and can only sustain maximal activity for a very short time. It's more of a rapid buffer than a long-term store.
So constant oxygen supply is paramount. What happens then during ischemia? If a coronary artery gets blocked and oxygen is cut off, how quickly does the muscle fail?
It fails remarkably quickly. If you induce stopped flow ischemia, say by occluding a coronary vessel, coordinated contractions cease within about 30 seconds.
30 seconds. That's incredibly fast. Is that because ATP runs out immediately?
Surprisingly, no. While ATP and creatine phosphate levels start to fall, they aren't completely depleted within those 30 seconds. The cessation of contraction seems to be triggered by other metabolic changes occurring rapidly, perhaps accumulation of metabolites like inorganic phosphate or protons, or changes in ion gradients, rather than just running out of ATP entirely at that instant.
Interesting. And what about recovery, if blood flow is restored reperfusion?
It depends crucially on the duration of the ischemia. After about 10 minutes of ischemia, creatine phosphate is very low, maybe only 20% of ATP remains. But if you reperfuse at this point, the heart can often restore its energy stores and regain significant contractile function.
Okay, 10 minutes is potentially recoverable. What about longer?
If the ischemia lasts for 20 minutes, ATP levels drop much further, maybe to just 5–10% of normal. Now, upon reperfusion, there's significantly less restoration of energy stores, and contractile activity is much more severely impaired, often permanently. This highlights the critical time window for intervention in events like a heart attack. Irreversible damage starts to occur relatively quickly.
A stark reminder of the heart's dependence on continuous oxygen and blood flow. Okay, finally, let's look at situations where the heart muscle itself changes structurally over time, cardiac muscle hypertrophy, and the group of diseases called cardiomyopathies.
Right. Hypertrophy simply means an increase in the size of individual cardiac muscle cells. The heart muscle gets bigger, but the reason why and the consequences are vastly different depending on the context.
So there's good hypertrophy and bad hypertrophy.
You could put it that way. We distinguish between physiological hypertrophy and pathological hypertrophy. Physiological hypertrophy is often called athlete's heart.
From endurance exercise, like running.
Exactly. It's an adaptation to increased workload, but it's generally beneficial. The heart muscle thickens or enlarges slightly, but this leads to improved cardiac performance, increased stroke volume, better oxygen consumption, and importantly, relaxation function is usually preserved or even enhanced. It's a healthy adaptation.
A stronger, more efficient pump.
Precisely. And interestingly, research points to factors like specific microRNAs, for example, miR222, playing a role in promoting this healthy, exercise-induced growth and even protecting against maladaptive changes after injury.
Okay, so that's the good kind. What about pathological hypertrophy?
Pathological hypertrophy is usually driven by chronic disease states, most commonly chronic pressure overload, think persistent high blood pressure or aortic valve stenosis, where the heart has to constantly pump against excessive resistance.
So the heart bulks up because it's overworked, but not in a good way.
Not in a good way. This type of hypertrophy is a maladaptation. It can manifest in a couple of patterns. Concentric hypertrophy involves thickening of the ventricular walls without necessarily increasing the chamber size, often an early compensatory response. Dilated hypertrophy involves an increase in the chamber volume, often with wall thinning later on.
And functionally, what's wrong with this kind of enlarged heart?
Both types typically show a decreased contractile response to beta-adrenergic stimulation, that's sympathetic drive we talked about. Their ability to ramp up performance when needed, their contractile reserve, is blunted. Dilated hypertrophy in particular is characterized by significantly impaired contractile ability even at rest.
So they're bigger, but weaker or less responsive.
Essentially, yes. And this decline in performance is strongly linked back to that dysregulation of intracellular calcium handling we keep mentioning.
It always comes back to calcium.
It really does in the heart. In pathological hypertrophy and heart failure, researchers find alterations in the levels, activity, or phosphorylation state of many key calcium-regulating proteins. The L-type channels, phospholamban circuit 2, the ryanodine receptors, the whole system starts to break down.
What are the molecular signals driving this unhealthy growth? How does pressure overload trigger hypertrophy?
It's complex, involving multiple signaling pathways. One important pathway involves calcium itself. Even modest, sustained increases in intracellular calcium, which can occur with chronic stress, can activate an enzyme called calcineurin.
Calcineurin.
Yes, it's a calcium-calmodulin-dependent phosphatase. It removes phosphate grooves from a transcription factor called NFA. Dephosphorylated NFAT moves into the nucleus and switches on genes that promote protein synthesis and cell growth hypertrophy.
So calcium signaling itself can directly trigger growth genes. Any others?
Another pathway involves CAM plus calmodulin-dependent protein kinase, or CAMK. When activated, it can lead to the activation of another transcription factor, MEF2, by promoting the removal of inhibitory proteins called HDACs, histone deacetylases. MEF2 also drives hypertrophic gene expression.
HDACs. Histone deacetylases, those affect how DNA is packaged, right?
Exactly. Which brings us to the role of epigenetics. And also remember that impaired beta-adrenergic response. That seems to involve the actual internalization and downregulation of the beta-adrenergic receptors themselves from the cell surface, a process involving other kinases like PI3K and beta-adrenergic receptor kinase 1, BRK or GRK2.
So it's a multi-pronged attack. Altered signaling, changes in gene expression, reduced responsiveness. Is there hope for targeting these pathways therapeutically?
There's a lot of research focused on that. Animal models like mice with surgically constricted aortas to mimic pressure overload are crucial for studying hypertrophy and the associated fibrosis, scarring. These studies have revealed that epigenetic modifications, changes in histone methylation or acetylation, play a significant role.
Modifying the histones around the DNA.
Yes. For instance, inhibiting a specific enzyme called KDM3A, a histone lysine demethylase, was shown to prevent hypertrophy and fibrosis in mice under pressure overload. It even protected against ischemia perfusion injury. Conversely, making cells produce more KDM3A-induced hypertrophy.
So KDM3A looks like a key player.
It does. Similarly, inhibiting certain classes of those histone deacetylases, the HDACs we mentioned, has also shown promise in preserving heart function and inhibiting hypertrophy in animal models. So KDM3A and HDACs are definitely considered potential pharmacological targets for treating pathological hypertrophy.
That's really exciting, targeting the fundamental gene regulation. What happens if you remove the pressure overload? Can the heart shrink back down?
Yes, to some extent. Pressure unloading can lead to cardiac atrophy, the opposite of hypertrophy. The heart mass decreases, capillary density might reduce, levels of proteins like SERCA2 can go down. This process also seems to involve signaling pathways like PI3K, suggesting it's an active, regulated reversal, not just passive shrinking.
Okay, so hypertrophy is about cell size. Now let's talk about dilated cardiomyopathy, DCM. How is that different?
It's a crucial distinction. DCM is a primary disease of the heart muscle itself, characterized by dilation or stretching out of the left ventricle, sometimes both ventricles. This dilation leads to impaired contraction systolic dysfunction, which results in heart failure. It also carries a significant risk of sudden cardiac death due to arrhythmias.
And unlike pathological hypertrophy, it's not typically caused by pressure overload.
Correct. While some cases can be triggered by inflammation, toxins, or other systemic diseases, a large proportion, probably most commonly, are caused by genetic mutations. These mutations often affect the proteins of the sarcomere itself or the desmosomes holding cells together.
So faulty building blocks.
Exactly. Mutations in genes for titin, myosin, troponin, phospholamban, tropomyosin, even sodium channels have all been linked to DCM. It's a defect in the fundamental machinery.
Can you give an example? Maybe that phospholamban mutation you mentioned earlier?
Yes. The R25C phospholamban mutation is a great example. It was found in a family with adult-onset DCM. Affected individuals had severely reduced left ventricular ejection fractions, meaning the heart pumped out very little blood with each beat, sometimes only 17–40% of the volume, and they suffered from atrial fibrillation or ventricular tachycardia.
Just from changing one amino acid in phospholamban, arginine to cysteine at position 25.
Just that one change. And when researchers engineered rat heart cells to overexpress this mutant R25C-PLN, they saw exactly what you'd expect to cause DCM.
What do they see functionally?
They observed a decreased extent of cell shortening, contraction, a smaller and slower intracellular calcium transient, decreased sensitivity of the SR calcium transport system, and importantly, an increased passive leak of calcium from the SR.
A leaky SR again, like in CPVT, but maybe through a different mechanism linked to PLN?
It seems so. The overall consequence was an elevated level of calcium in the cytosol during diastole, during the resting phase. This elevated basal calcium is thought to contribute significantly to the development of arrhythmias in patients carrying this mutation, on top of the weakened contraction causing the heart failure.
Wow. It really drives home how a single molecular defect can unravel the whole system, leading to devastating disease. We have certainly gone deep today. I mean, from the microscopic structure of a single sarcomere right up to the complex signaling cascades that govern every heartbeat, and how the heart adapts or tragically fails to adapt to the demands placed upon it.
You should now have a much clearer picture, a real mastery of why your heart can't afford to fatigue like skeletal muscle, how it so cleverly manages calcium to fine-tune its force beat by beat, and the incredibly intricate ways it responds to everything from a jolt of excitement to subtle genetic variations.
Yeah, this isn't just abstract theory, is it? It's the absolute foundation for understanding heart health, heart disease, and how treatments, current and future, actually work at a fundamental biological level.
Absolutely, it's the core knowledge.
So, to leave you with something to think about, consider that remarkable balance the heart maintains. It's this constant involuntary pump, working nonstop, yet it's capable of these extraordinary minute-by-minute adjustments in force, rate, and relaxation. Given the deep insights we've uncovered today into its fundamental cellular and molecular workings, what do you think might be the next frontier in understanding and treating cardiac conditions? What new questions does this mastery raise for you about the resilience and vulnerability of your own heart?